of diphenylamine, bromelain, and citric acid through computation. Next, index of the project. So we will learn first introduction, then objectives of the project, then methodologies used, then main findings, then conclusion, and lastly references. So let's begin with introduction. So we have to know first of all what is bioactivity. Bioactivity is defined by obtaining specific effects after exposure to a particular substance. These effects may include tissue uptake, metabolism, or physiological response. Tissue uptake means an instance of absorbing. Metabolism is the chemical reaction in the body cells that change food into energy. Physiological response is the refers to the emotional, cognitive, and behavioral reactions. So next we will move what is meant by bioactive compounds. A type of chemical found in small amount in plants and certain foods such as fruits, vegetables, nuts, oils, and whole grains. Bioactive compounds have action in the body that may promote good health. Bioactive compounds are those food components that have an effect on the body as a whole or specific tissue or cells. Example, flavonoids, caffeine, creatine, polysaccharides, and prebiotic. These are the major food bioactive compound sources and its classification. First, plant, animal, fungi, and bacteria. So next, bioactive compounds are chosen. I have chosen three bioactive compounds first, namely diphenylamine. Diphenylamine is an organic compound with a formula
Yeah, I
The
think
compound
Mousumi,
is
you're again having
derivative of
an internet issue.
aniline consisting of an amine bond to two phenyl groups. Hello? Hello, ma'am?
Yes, yes, Mousumi, you're audible, audible.
Hello? Ma'am, can I start? Hello?
Yes, yes, start,
Hello?
start.
So uses of diphenylamine. Diphenylamine is widely used as an industrial antioxidant, diamordant reagent, and also employed in agriculture as a fungicide and anti-helminthing. Anti-helminthing is an agent that destroys parasitic worms. Diphenylamine is a useful intermediate for the synthesis of rubber, chemicals, medicine, dyes, etc. So next compound is bromelain, an enzyme found in pineapple that breaks down other properties such as collagen and muscle fiber and has anti-inflammatory properties. Collagen is the most abundant protein in our body. Muscle fiber is help to control the physical forces within the body. An anti-inflammatory is a drug that used to reduce inflammation such as redness, pains, swelling, etc. Ipacine 6-bromo 5-chloro-3H 1,3-benzoxyl 2-one. Uses. It is used as meat candidizer in the body food industry. Bromelain is used for reducing pain and swelling, especially of the nose and sinuses, gums and other body parts after surgery or injury. So next compound is citric acid, a pleasantly sour-tasting organic acid with chemical formula C6H8O7, obtained especially from lemon and lime juices or by chemical breakdown of sugars and used as flavoring. Citric acid is a naturally occurring acid. It is found in various fruits and vegetables. Example, lemon, which has the highest amount of citric acid. Ipacine, 2-hydroxypropane 1,2,3 tricarboxylic acid. Uses. Citric acid is used as food as flavoring agent and preservative. Due to its sour taste, it is used in making certain candies. It is used in processed food products like beverages, soft drinks, etc. So we will move objectives of the project. First, we have used ChemDraw and Pass Online software to analyze the bioactivity of the chosen compounds. Second, we have done the literature survey of these compounds and explained them in this project. And third, we found the following useful bioactivities of the compound. First, diphenylamine and their bioactivities. Cutaneous inhibitor, protein kinase inhibitor, glucose oxidase inhibitor, fibrinolytic aldehyde oxidase inhibitor. Next, bromelain and their bioactivities. Peroxidase inhibitor, histidine kinase inhibitor, antioxidant, antiplastic, and vasoprotector. Just citric acid, glutaminase inhibitor, lipotropic, dextrin inhibitor, antisebric, and lysosostapine. So we will move methodologies used. So what methodologies used? Step first. First, we have used ChemDraw to draw the chemical structure of our fusion compounds and save them as the MDL mole file. Second, next we have registered on the way to drug pass online website and predicted the bioactivities of the fusion compounds. And lastly, we have done a literature survey to explain the bioactivities of the fusion compounds and explain it in details in the next section of this project. These are the steps we have followed in the methodologies. So we will next move main findings. So first compound is diphenylamine and their bioactivities. First, cutaneous inhibitor. Cutaneases are inducible extracellular enzymes secreted by microorganisms that are capable of degrading plant cell walls. Inhibition of cutaneous has been shown to prevent fungal infection through intact cuticles. Second, protein kinase inhibitor, a substance that blocks the action of enzymes called protein kinases. Blocking certain protein kinases may help keep cancer cells from growing. Some protein kinase inhibitors, such as imatinib, vemurafenib, and giftinib, are used to treat cancer. Glucose oxidase inhibitors. Glucose oxidase inhibitors are a group of antibiotic drugs used for management of type 2 diabetes mellitus. The classical alpha glucooxidase inhibitors currently used for TDD treatment are acarbose and voglibose. Acarbose is a oligosaccharides produced by fermentation of bacteria, and voglibose is an alpha glucose oxidase inhibitor that is used for lowering postprandial of glucose blood level in people with diabetes mellitus. Next, fibrinolysis. Fibrinolysis is a normal body process. It prevents blood clots that occur naturally from growing and causes so blood clots means it is the natural part of the body defense mechanism. Fibrinolysis is a highly regulated enzymatic process that prevents unnecessary accumulation of carboxylic acid. So next, bromelain and their bioactivities. First, peroxidase inhibitor. Peroxidase inhibitor is the inositol phosphoglycan-like compound. Is the general inhibitor of peroxidase and catalyze involved in the modulation of hydrogen peroxide level that acts in different metabolic pathways as a single transducer. Next, histidine kinase inhibitor. These compounds generally inhibit by binding to the 4-helix bundle. 4-helix bundle recurring structural motif in protein that contains the conserved histidine residues, thereby causing aggregation of the HKs. This kind of inhibitor usually prevents bacterial growth by non-specific mechanism independent of HK inhibition. Next, antioxidant, a substance that protects cells from the damage caused by free radicals. Antioxidants are mostly found in plants, foods, etc. They are natural molecules that help neutralize harmful free radicals in our bodies. Bromelain's anti-inflammatory and antioxidant properties were found to protect the esophagus and tongue tissues in cursive bones. Next, antinoplastic. An antinoplastic is any drug used to inhibit the growth and spread of cancerous cells. Antinoplastic are used in chemotherapy to kill cancer cells and prevent their spread. Antinoplastic combat the development of cancer.
Yeah, Mousumi. Yes,
Hello.
yes.
Hi, ma'am. The next citric acid and their bioactivities. First, Glutaminase inhibitor selectively and reversibly binds to and inhibits human glutaminase, an enzyme that is essential for the conversion of the amino acid glutaminase and into glutamine. Glutaminase can be used in the wheat flour, bakery products, pasta, and some flavoring preparation. Next, lipotropic, having an affinity for lipids and thus preventing or correcting excess accumulation of fat in the liver. Lipotropic compounds are those that helps catalyze the breakdown of fat during metabolism in the body. Lipotropics are necessary for maintenance of healthy liver. Next, dextrinase inhibitor. Dextrinase inhibitor is a member of the glycoside hydrolase family, 66, and produces isomalto oligosaccharides of various sizes by intermolecular transglycosylation. They are commonly used as anti and retrograde tracers in neurons. Next, anti-seborrheic, preventing or relieving the symptoms of seborrheic or seborrheic dermatitis. Topical anti-seborrheic agents are class of drugs used to treat seborrheic dermatitis, acne, and dandruff. Next, last, lysosostapine. Lysosostapine is an antimicrobial agent belonging to major class of antimicrobial peptides and proteins known as the bacteriocins. Lysosostapine is used as a research and diagnostic tool. Next,
Yeah, Mousumi, am I audible to you? Yes, I think now she has again joined.
conclusion, there are many opportunities that diethylamine, bromelain, and citric acid present on the market, but these are not without complications. From this overall project, I have learned bioactivities of the chosen compounds and their chemical structures, and this. Hello?
Yes, yes, you're audible. Go ahead.
Hello? First and enlighten us to prepare this project. Thank you so much.
Okay, Moshumi, thank you. And it was really nice. I would